right? We're just going to say, oh, well, here at this membrane, here's my phospholipids, right? At rest, nothing is happening. All right? And when I called nerve and muscle excitable, what I was telling you is I can change that situation. And that's what your authors are telling you, that this situation can change. So this ionic distribution or difference exists in most all your cells. But most of your cells aren't going to have an ability to really change that. I mean, half of them, we should be honest. So we don't call epithelial tissue and connective tissue excitable. We call muscle and nerve excitable, okay? So this would be our situation, right? And this comes down to, and here's all, again, to try to really, again, make this, it's not a simple, again, it's not a simple concept.
Which is why the questions were problematic for you when I was trying to understand how it doesn't take into account things like the electron transport chain and how that changes the...
It won't. No, it won't. Nope. Because that's well and truly inside the cell. That's not going to change the membrane.
Well, there's nothing that's
not a factor in it, okay?
One's not just like positive and one's not just negative on the outside.
Okay, this has to do with leakage channels and pumps. Mm-hmm. The electron transport chain is happening in here, and it's doing all its transfers right inside the cell. And still, ultimately, we're going to have a balanced charge inside the cell. Okay? So, again, this concept comes about because of, in muscle and nerve, because these tissues have specialized transporters, right, that we're going to call channels. They're gated, the potential's gated, we're going to call them channels, and pumps. Right, so we're going to talk a little bit about what those are, and I'm going to explain, hopefully, how this happens. But here's what I want you to not forget. That when we talk about excitability, whether it's muscle or nerve, we focus on just 3 ions, 3 really important ones. I'll bring up a 4th one, but it's, again, when we're talking about normal function, right, we're talking about sodium, potassium, and calcium. These 3, all cations, right? Anions often go with these, right? So sodium will have a chloride, so we will mention chloride as well. Because I said positive and negative, they pull each other. There's an attractive force, because opposites attract, just like we talked about when we talked about the organization of the atom as a nucleus and electrons orbiting. All right? So if we took any cell in your body, again, any cell in your body, we just have the neuron in front of us, and we're going to focus on the neurons. Here's what you'd see. We talk about what was intracellular and what was extracellular. So inside, right, so if we complete this, and I said this is just a simple drawing that you want to think about, right, and we complete this all the way around. So inside, and of course my color chalk probably will erase. Here's the situation. Right, we'd see lots of potassium, don't forget that, right? We would see some magnesium, which is actually positive as well, right? We'd see phosphates, which are negatives, and sulfates, that might be -2. Sulfates, -2, right? We'd see these negatives and positives, and then we'd have a lot of protein, and proteins are often charged as well. So we have this charge distribution issue, right? So that's inside. And I will get you, if you go on to A&P2, you're going to tell me this in chapter 21. And I'll say to you, you learned this in A&P1. Now outside, it's sodiums, it's chlorides, it's bicarbonates, it's calciums. Now, these are inside as well, but much lower. So I'm giving you I'm showing them big because there's a difference in their amounts. So there's a concentration difference. So this is a problem, isn't it? Because we said, well, cells really like equilibrium. That's what we taught you in diffusion, right? But the problem here is those are charged and they can't pass through this membrane. Okay? So here's where we want to just begin to understand the cell. So extracellular and intracellular are not the same composition. But again, if we counted up the total number of negative and positive way outside, right, out here and in here, they would actually be equal. They are not equal at the membrane. And so at the membrane, we see this. When nothing is happening, that's what we see. Okay? So that's where we want to start, because a couple of those questions we talked about, I mentioned all the potassium inside, or all this. It's not. That's not the way it's going to be. Okay? And we're gonna go through it again and look at those channels now. So I left off looking at this idea of function, and I asked you guys, please, it was one of your questions here, question 3, and I'm very serious about this. Apply this idea to anything you do, right, to understand neural pathways. So this also is a good demonstration of, remember that action potential, right, you reach threshold. We talked about that in chapter, right, 10. So there's a minimum requirement for change, because that's what we're after. We've got to change this situation if we're going to get something. And that something is an action potential, because that's our electrical communication. Okay? So I just have to embrace that thought to get what we're going to talk about here. So minimum requirement of change, threshold, and then I get a lot of change. And I spread that change down that neuron, right? And that spread I call propagation. Fair enough? I mean, so just because we're going to use these terms. Amount of change then depends on the strength of the stimulus. Okay, I'm going to give you one that's really easy to embrace. All right? You ever come inside from being outside, warm night or morning in Texas, and you start scratching? You've been bitten by a mosquito. You didn't even know it landed on you. Okay. Well, the mosquito has evolved techniques to not be swatted, right? Which includes, it's very soft. You ever watch how they spread out, distribute their weight with those long, long limbs? Yeah, that's not just a fluke. That works really well, because you didn't feel them. But I didn't scratch thinking about it, right? Right? You didn't feel them. Shame on you? No. It's just, this is what we're talking about. So that mosquito landing on you did not, right, cause enough change for these guys to detect. And you were not aware because you never sensed it, did you? Now, there are many times when the mosquito bites me and I feel that, right? And I swat them immediately. So they caused enough change in my skin. They made a threshold, right? And we say the stimulus is going to be called a graded potential. So when you read that, so you're not confused, right? Amount of change depends on the strength of stimulus. So when he's landing really, really soft on me, there's not enough change to generate the stimulus, right? But then when I start to swell, then I know, because there is enough change at that point, right? And then when the chemicals he's released into my arm, right, create the itching, yeah, there's enough change, right? So other times when you felt that land on you, they had to reach threshold. So that's what we're looking at. Here they give you an example of feeling the temperature of water. You can apply this to anything. Because we are what goes on up here, aren't we? And if we're not aware of it, it just doesn't happen. It's not our reality, is it? Let's face it, this thing up here creates our reality. And that reality is shaped by your sensory. So we want to see these pathways going together and for the rest of the semester, that's what we're going to be dealing with. But in this chapter, we really need to talk about this. All right, this change. Because if I don't get this changed, everything else just doesn't make sense. All right, so this just says, okay, what next? At the end of this unipolar, because that was a sensory, so coming in here to the dorsal root ganglia, right, where the cell body were, as it comes in its synapses, well then, to continue the change, I need a neurotransmitter, right? I say in this chapter says to you, there's a few examples where the neurons can directly electrically excite each other, like you guys look right now, right? Except, right? But the rule is usually that you'd have to pass a chemical between this space. So if David wants to talk to Langston, there's got to be a chemical released. I don't know what that is, David. Right? So, right? But we call that a neurotransmitter. Right? And we have all kinds of different classes, chemical classes, but they really tend to be some derivative of proteins. Small proteins, peptides, amino acids. Okay? they cross the synapse, and this is what we picked up, right? This is very familiar to us because that's what we talked about in the muscle, right? We said we got to the end of this motor neuron, right? The axon isn't direct communication, so it releases acetylcholine. That's its neurotransmitter. And acetylcholine is still going to be used for us here in this chapter and in the following chapters, and that crosses and causes, right? In your case, it was a new muscle. I don't get that out of the way. We're all past that, right? So, right? Instead of, now we're going to replace that muscle with another neuron, right? And we're going to say, yeah, this starts an electrical change. But we showed you the electrical change in the muscle that occurred. And at that point, right, well, what did we do? What happened when acetylcholine bound to its receptor on the muscle? What did we do?
The channels open.
Which one specifically, Tanner? Take a guess. We have our big three.
Sodium?
Yes, right. So sodium, because sodium's high outside. And what we're going to do, we're going to say it depolarizes, because this is polar. And in fact, again, I said if we put that electrode in there, it's a minus 70 millivolts polar. It's actually quite a bit when you think about all the cells in your body, right? You got a lot of potential, right? So, yeah, you started opening sodium. So we're going to do that here as well. We won't always do sodium. In the muscles, it was always sodium. Okay? So I want you to think right now, what happens if I open a potassium one with that neurotransmitter? Let's just think about that for a second. Well, it's high in here, low out here. What's going to happen? Potassium leaves. What's going to happen to this difference? I mean, you all need to just grasp this because it's not hard.
It's going to be more negative.
It's going to be more negative. And what are we going to call it? We say it's hyperpolarized. It's more polar. And that's a problem because that means it's going to take more change to get a response, isn't it? Because my threshold is still the same. And if you want a number, your threshold is usually minus 55. Somewhere around that area. What if I open a chloride channel? What happens then?
It'll become more negative on the inside.
Again, yeah. So really the same result, even with a different channel, right? So what I'm trying to illustrate here, and Katie's really helping me out here, is, and I hope you all get this, right, is that I'm not going to have an infinite amount of choice, right, in what's going to happen here. But this will either say you're going to stimulate, right, the change, or propagate the change, action potential, or you're going to inhibit it, right? And so when I'm in pain, I'd really like to inhibit it. Wouldn't I? Right? I want you to think about logical things that you would experience. Pain, right? You have to sense that. It creates a sensation up here. I probably want to inhibit that, right? So the drugs we use, of course, are going to try to do that, aren't they? Right? If I want to prolong the response, well, then I want to stimulate, right? So there's very practical applications, even though this is really electrophysiology. Yeah, Katie? Mm-hmm.
Whenever my little brother was like 5, he lost his toes and was like in the hospital, and they put an epidural in his back to prevent the pain from reaching his brain. Does that kill all feeling, or do they?
It probably did quite a bit on that area because of the pathway that they selected. But yeah, because it was so low, it probably had a pretty wide effect on his body. And again, we don't really care about those side effects medically. We just, again, we don't have a perfect system of pharmaceuticals and system of delivery. We just want to make sure that he's not experiencing pain there. So yeah, you're going to see some effect in other places that weren't intended because it's not a perfect delivery, right? We're just stopping it from going up, and that's going to stop a lot of things.
What is it? How does it do that, like chemically?
Well, that's what we're gonna talk about here. It's really it's ultimately comes down to it's going to change what happens at these membranes and the ability to transmit, right, ions, right, so that you can't then have an action potential. Because the action potential is the communication between these things. So if I block, right, using some other chemical that looks like a neurotransmitter, like acetylcholine, yeah, I can block its binding to it, well then the muscle doesn't do anything, does it? That's how curare works, right? What South African hunters use as a poison, right, that induces paralysis. It binds, it looks very similar, binds the same place that acetylcholine would. Acetylcholine can't there, so there's no sodium gates that open, there's no depolarization, there's no action potential. Your muscle's doing nothing. So there's lots of different mechanisms, because neuroscience is a fascinating field. We can block it many different ways as we talk about this sequence. I mean, ultimately, if I block the calcium from leaving the sarcoplasmic reticulum or binding to the troponin, I can get really the same effect, honestly. So there's lots of different ways that we can achieve this, but that's what we're after when we treat things, like pain pharmaceutically. We want to prevent that change. And we're using change very broadly here, but change in electrical potential. All right? So, and again, this goes on. You don't have to write this down. This is clearly just, I'm saying we're going to follow the pathway. So that's why I said we're going to start here in chapter 10, and we've been talking in chapter 10, and I'd like to get 12, and get mostly through it, all right? In as much as we want to just give you this foundational way that neurons talk to each other. But we don't want you to forget why this is important, as we've been talking, and I've tried to give you an example here, right? So I had mentioned to you already that your sensory cortex is in your parietal lobe for your general senses. Your special senses are scattered in the brain, all right? And so this, right, you will get this sensation go up there, right? It usually synapses in the thalamus, and you guys will be studying the brain in lab very soon, if not, if you haven't already started. It'll have a synapse in the thalamus, and then the thalamus relays it up to the right area in the parietal lobe. And then, like I said, if you want to do something about it, like they give the example of the temperature is not right, and you want to change it, well, then I have to send a motor, right? Action potential. And so we want to remind you that that's a somatic division, because that got missed on the test too, or excuse me, the quiz. Because somatic, I'm going down to the skeletal muscle. If I'm using that, that's my response, right? My efferent fibers, my response, and there, the somatic implies I'm going to end up talking to a skeletal muscle. So I want to be very careful about who responds, right? Because when we talk about the autonomic, not in this chapter, but in chapter, I think, 15, I have to talk a little bit, I won't talk about the whole chapter, but when we talk about autonomic, well, then those effectors are cardiac and smooth muscle, okay? So, and I say there's just these upper motor neurons, so they're higher up, that's what they mean by that. In the precentral gyrus, that sounds probably like a bit of a gibber right now, but when you look at the brain, well, the central gyrus is this right here. So in front of it. Central, that's all it means. Precentral gyrus, right there. So would be in here, would be where your motor action potentials would begin. If I want to change the temperature, that's where it starts, okay? Moves down the spinal cord, right, because that's the conduit. Why they select that conduit. And, right, lower motor neurons, so there's other motor neurons in. Yes, and now that you've studied the spinal cord, you know that the motor neurons are actually going to be in the anterior, right? And then they're going to go out. So there'll be a synapse in there, long axons coming down. Those synapse with something in the anterior horns of the spinal cord. And that's going to tell, as I say here, your supinators and pronators to turn that knob, all right, so that you change the temperature. And I said, just practice this, because I'm sure you bathe fairly often. I know we've had that discussion. Alexis isn't here today, right? But we did talk about that when we talked about skin. But even if you don't, you probably turn water on a lot. I mean, that's just one way to think about what's happening here. All right? So here is where I started, you guys, because that's where the rest of the chapter is really going to deal with. We have to understand that change. All right? So, outside membrane positive, inside negative, and again, we said at rest, when there's no energy transfers occurring at the membrane. There's always energy transfers going on inside the cell, but at the membrane. All right? So a cell membrane is polarized. So this is very nice. It shows you, again, it's a much better drawing than my drawing up here, but the same idea. Just draw those charges all along there. All right? Plus, 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 negative, negative, negative. I said key ions, like you told me, Tanner. Sodium, potassium, calcium. From here and the rest of your life, understand that sodium is high outside of a cell and potassium is high inside, right? And I say, right, this polarization is from differences in concentrations and the permeability of the membrane. What is it going to let through? Okay? That's really key here. But this is stuff really you already got when we talked about muscle in chapter 10. Okay? So what are we going to do in chapter 12? Well, we're going to name some of these channels. We're going to tell you, look, it's not just this one-size-fits-all sodium channel or gate, right, you're going to see some groups of these. And we want to think about what they mean. Now, when I say think about it, it doesn't necessarily mean that in fact, I didn't ask any quiz questions on the different types of transporters in the membrane. And I don't see myself really making that a big point. It's an important part of this chapter, in as much as you want to think about, this gives you a lot of choice. All right, it gives you a lot of choice in what you do. The more structures you have in that membrane, the more, right, capabilities it has. Okay, so and so this will also help you when we start talking about senses, right? Because how do I start? As we talked about that mosquito, how do I actually start that action potential in those Meissner's corpuscles? How did I start that? Because it has to start somewhere, right? I'm going to talk about propagating it, moving it, but it has to start somewhere. How did I get a change? How did that mosquito change the polarity of my membrane? Because that's what has to happen. Because when I didn't detect it, it didn't. So how does that happen? Well, that happens because there's mechanically gated channels. They respond to changes in physical location, like rubbing. Right? When we get into our special senses, we'll talk about light being the stimulus for your retina. Specialized gates, right? It's still going to require movement of these, but we're going to have a lot of variety, and that's what gives us variety. A lot of variety in our transporters, okay? So, fundamental, again, don't ever lose this. Okay, you don't have to remember the phosphates and sulfates and everything, but potassium, sodium, calcium, don't ever forget that. The rest of your life, please. If you're going to stay in health, keep that. All right? And know again that this is a permeability. There's changes in permeability that are going to happen. All right, so when we move these ions, it's going to be electrical change. All right? And we move them by, again, we broad category ion channels. All right, so here's a couple. This is just a nice cartoon. It says, okay, here's your muscle one that you were talking about, Tana, right? Here, acetylcholine comes in, binds to its receptor, and when the receptor binds here, it opens the channel, right? And so we can have that channel ions moving in, ions moving out. Again, we've got sodium, potassium here, calcium like they should. So it's a great cartoon, very simple but good. All right? So when that channel opens, if it lets all of them through, okay, and that's not what happens in muscle, but if it lets all of them through, they say, well, look, the concentration gradient says that these two will move in, because there's more of them out here than in here, and this one will move out. All right? So it turns out that these channels, yay, are actually specific, right? They don't do, right, just let all three pass through here. So the channels are specific based on the charge of the ion, the size of the ion, and you're going to only interact, sodium, calcium, potassium in this case, right, with a specific channel because of these characteristics of you as an ion. So you're the right size and or charge. Some are opened by specific events, and we call them gated. So like you would have a gated community, you're choosing what you let through. So that's when you see that word gated, that's what they're talking about, a specific event. Right? I'll be with you in just a second, Kale. Ligand gated, which is what these are, because this, right, that's a chemical. When we say a ligand, we're just basically a broad term for a chemical. Some chemical, some molecule. We also, your book refers to this as ionotropic. Okay? I won't probably use that word, honestly. There's a couple words in this chapter I'm not going to use because, again, it's an introductory class. We want to give you the basic rules. But these ligand gated open, right, when a signaling molecule, acetylcholine, binds. Okay, so that's our rule. So it's gated because there's a control. And this is what we saw with muscle. What was your question, Kale?
With how how do steroids affect this? In the sense, like, if you have too much like calcium that moves into the membrane.
Okay, I'm just gonna back you up a second. A steroid isn't gonna need a channel gate. Okay, it's gonna move right through here, and it's going to then bind to the DNA, most likely. All right, and it's going to cause the DNA to specifically make a certain channel.
Yeah.
And then the cell will put that channel in the membrane. So the long-term effect is a steroid could affect the polarity because of that.
Yeah.
Okay? But yeah, it's going to move right in because of its chemical nature.
Yeah, because you have calcium being brought in by D.
And so it's going to bind to the DNA and get more of these made. Because this is not a constant, right? Cells would make these and replace these on a regular basis.
They'll physically open the gates then if you have too much inside the cell?
Well, here's the reality, is it could be a gate that would just stay open or be a leaking channel.
Yeah.
And in that circumstance, that'd be good enough to let the calcium pass. Okay? But I don't know specifically, I'd have to look at which gate they actually put in, but that's the mechanism, that steroids just enter right on in, and then they reprogram the cell's nucleus to produce more of these, because these are proteins, right? These everything we talk about here that's not a lipid, that's probably going to be a protein.
That's what I was trying to figure out how you deal with like non-polar or closer non-polar ones that can go through the phospholipid bilayer. How do you deal with them with electrical impulses if there's no way of regulating physical pressure?
Okay, it's just that there's a whole variety again of them that responded. So here I'm just giving you a few here, and I don't this is not the point of my chapter. It's just to say, look, you have a lot of variety in your membrane. So you do need to be aware of this because ligand gating is not the sole thing, all right? So here's what I was talking about with the mosquito, landing, physical distortion of the membrane. So when they land and there's some pressure, you distort the membrane. When we're talking about touch in general, that's a physical distortion, right? Pressure is a physical distortion. So in those those sensors, like the Pacinian corpuscles, or the Ruffinians, or Ruffinis, and Meissner's, this is what would happen to start the action potential. You're not relying on acetylcholine or another neurotransmitter, a ligand. You're changing, just by this effect, these membranes. So we say they're mechanically gated, right? And when they are rubbed, then they open, and again, knowing that sodium, calcium's high and outside and potassium's high inside, they'll let things through. And in this case, again, like Kayla was asking, they may not be so specific as well. Right? We might see. It just, I'm sure it probably depends, and you have a lot of these scattered in your skin. All right, or we have voltage gated. All right, so you're going to see the ligand gated and the voltage gated in this chapter. Okay, so the voltage gated, it's kind of a cute little ball and chain example I give you there. It's a nice cartoon to get right. If the electrical properties of the membrane change, because that's what we said was going to happen with acetylcholine. Bound, aren't we? If they change, then these channels can open. All right? So here again, just the reality that we had a ligand-gated one that was somewhere down here, and when it opened because of acetylcholine, it changed the potential, and that change of potential triggered the opening of this one. So what you see is a lot of cause and effect, but that's the way the body works. And all you got to figure out is, how are they related to each other? First you learn the vocabulary, and then you figure out, okay, I get it now, right? This one influences this one, and then I get this. Okay, so we will talk about our voltage-gated channels along, especially all the way down this axon. That's what the majority of them are going to be. God bless you. And that's why I said when we talked about myelin, you needed just this little bit of change, close enough in space, spatially change, and time, of course, space and time, that you would cause another gate to open just a little bit further down, and a little further down, a little further down, okay? So you get this kind of cascade, don't you? Like the start of an avalanche almost, right? But not as deadly, right? Hopefully it's life. All right, and then you have these, and this is what helps with the with that resting membrane potential. Is that in those neurons, right? And I don't think it's true for muscle at all, but in the neurons, I don't know an example. Well, actually, it is in cardiac muscle,
Yeah.
not in skeletal muscle, right? You would see leakage channels, all right? And leakage channels do just what they said. They have a pattern of opening and closing. It's not random, but they flip. It's like, right? That kind of thing. They're flipping. So intrinsic rate, again, so they have a pattern unique to the gait between open and close. And this is really important for resting. So I might ask you a question like that. I might say, well, how do we establish resting membrane potential? That was one I had on the quiz, and I went ahead and took it off because lots of students missed it, and I don't think my choices were clear enough in the question, so I did take that one off. All right? So open and close. So we would see these, again, we could see any of these in the membrane of the cell. We're just going to look at some specific examples. So here's our membrane potential. Again, that's exactly how we would do this, right? We just put a very small probe, right? And we see on average, sometimes it's a little bit lower, but for the average cell, it's about minus 70 millivolts. All right, and this is stored electrical energy. That's why it can make change, right? Because energy is the capacity to do work or make change. That's the dry definition of energy. Okay, so we're just showing you that, yes, this is not something we're making up. It's really, you can actually measure this, right? And it really is only at the membrane surface. And because we can change this situation in muscle and nerve, we say they're excitable. All right. I'm sorry if I go too fast for anybody. Just let me know. All right. I want to get you through this in essence. So that part is honestly not that important. It's just the idea that yes, we know this is minus 70. Now, if this cell stayed at rest, that's what you would see, right? This is time, a very short time, by the way, right? This is in milliseconds. But we would just keep on, right? If nothing was happening, nothing was changing, we'd just keep on seeing that change, that same, no change, right? Same potential. And remember, potential is a capacity, right? All right? So I say, when it's not being stimulated, these ion channels are closed, right? Except for the leakage ones. So the ligand-gated ones, the voltage-gated ones, right? The mechanically gated ones, they're closed. Sodium is about 10 times higher outside than inside. Potassium is just in general higher inside. I have to look at that. Right, so we have negative proteins. Cytosol has, again, that's the stuff inside the cell. So the inner membrane has negative proteins associated with it. The cytosol has sulfates and phosphates, all negatively charged and associated with the inside. So we get this, again, negative. Negative, positive, with no change occurring. So please understand what we have here is we're following change. We're following change in electrical events, millivolts here. But right now, we're right here. So, all right. So our action potential, right? We want to do something, as the name implies, because resting membrane, you're not doing anything, right? So action potential is going to require the change in this that you've already been exposed to, all right? So again, this resting would remain and nothing would happen unless there is something that occurs, some change that occurs. Sodium is key, as we talked about, right? So when a channel for sodium opens, the concentration gradient, this difference, that's what we mean, is going to drive sodium in. Sodium is going to enter, all right? And so there's a voltage change that I mentioned. And that voltage change will end up triggering these voltage-gated channels, voltage-gated channels. And we would say, as sodium entered, and you guys observed, that the inside would become less negative. And so we'd represent it this way, wouldn't we? If the inside is becoming less negative, it's moving towards zero. Okay? So we call this, all of it, as soon as we started to become less negative, right, compared to at rest, you are depolarizing. If it becomes more negative, that's what Katie brought up, about potassium or chlorides, then it's hyperpolarized. Okay, but right now we're going to follow an action potential happening, and that would be a depolarization. Right? I say that it happens very quickly, but there's so much sodium outside this, and as these gates, these should be, if we had a neurotransmitter, which is generally how it's going to start, the neurotransmitter is going to be ligand gated and it's going to open a lot of sodium. Right? So it so much occurs that we actually overshoot. So for this brief period here, right, the inside of the membrane becomes positive relative to the outside, right? So here they'd be equal. There would be no charge, right, if it was at zero. But they actually, so much opens in such a short time, just know it becomes slightly, right, positive, or it's positive, not slightly, it's positive inside compared to outside. All right? Which is not a big deal. I'm going to tell you right now, no matter what you do, you're not going to go above this because of the mechanics of these gates in the situation, right? So that's why we refer to the response, because this is a response of the neuron, right? It's responding to change, the binding of acetylcholine. We say the response is all or none, because you can't send a stronger stimulus and get more change. It'll always top out at positive 30. So what happens to make a stimulus more intense, like that light in my eyes right now, right? Or if my voice is annoying to you, what if I grabbed you really hard, which I'm not going to do because that's a violation of, right? So what is it that would lead to your ability to detect that was a different stimulus, a difference in potential? Well, you'd send these more frequently, right? You'd send them more often. And that's what happens in pain, right? You just keep sending, sending, and sending. And later on when I talk about senses, I'll tell you that pain is a problem because pain actually never stops sending on its own. Your smell does, and your taste does. And we say that those senses adapt to their stimuli, right? They adapt. You know this because you might come in a room and go, God, David, what did you eat, right? And then, no, right? So, but after a while, I wouldn't smell it, would I? If you had some strong garlic. But after a while, I wouldn't be able to register, be aware of that because in my nose, the resetting of these channels is very, very slow. So we say you've adapted to that. And the same thing happens to taste. Like you taste something really, really spicy and you're like, oh my gosh, right? But after a while, you can keep eating it, right? Because you have these receptors, again, you've adapted. They're very, very slow comparatively to in resetting. Yeah, Kale?
Does that overactive, like when you have like what overactive mind, how it does I guess when it's saving information and going back and forth and it's happening faster, does that actually make the peripheral nervous system any more sensitive or anything like that outside of the central? Like does that?
It probably could.
Have,
it probably could. I don't know specifically, but it probably I would hypothesize it could.
Yeah.
All right. So we got our change, right? Sodium channels have opened, right? And so many that we've actually made it positive inside relative to outside. Well, the next thing, again, this change, right? I said everything's about cause and effect, leads to this change, which is due to potassium. So now potassium channels open. Potassium channels are slower. Sodium channels are very quick, so they actually could have started responding somewhere about here, but it took them a little bit of time, but they'll start responding, right? And they're slow to close, so you often hyperpolarize as well. So potassium channels, they'll open at this point so that I can do this, all right? It's very high inside, very positive, so it starts leaving. And as Katie, when I asked that question, said, this will become negative again. And so we will say the membrane is repolarized. And we specifically measure it to make that statement, right? Because the depolarization and repolarization are really, they're a change, and we're measuring the amount of change. So potassium gates open, potassium starts leaving, the inside starts to become negative again, right? You can when I say what started all these channels opening, we began with acetylcholine, right? But we know now that it could be a number of different stimuli, couldn't it, in sensory. But once we start the change, and it's enough of a change, threshold, we're going to send it. Unless, like Katie asked, we block it somehow, block that transmission. Because it gets to the end of a neuron, right, and it's going to have to, right, release a neurotransmitter to tell the next neuron in the pathway. And then it'll travel again, and the same thing will happen. So, right? And so I say again, recall the different channels we discussed, because that's really the initiation of these changes that we have going on here, all right? So, threshold, and we mentioned this, I pulled this figure actually from your old text, because it was good, because what you saw here is a very simple, this is really a simple concept that often just kind of gets overlooked because it's you don't see it. You're not aware of it happening, all right? You can't be, because if you don't make threshold, you don't have awareness, all right? So what physically, what is that? Well, so here's all these different axons, right, that could chemically communicate with this neuron, okay? Now, in this scenario, 2 and only 2 are sending a neurotransmitter. We'll stick to acetylcholine for now. And it's opening sodium, right? Ligand-gated sodium channels. And sodium's entering. But the change is not enough, because threshold requirement is minus 55. Okay? Well, if we get this other, right, neuron to release its neurotransmitters and therefore influence more sodium, we make threshold. And that's really all it is. You have a minimum requirement of how much sodium needs to enter so that there is enough of a change. And if you don't make threshold, which is an example of trying to block something, you don't experience, you're not aware. There's nothing that makes it up to the brain or makes it past that point. All right? So threshold is, frankly, a minimum requirement. Right? So I say here again, initially this depolarization, because that's what's happening here. This is still depolarization, isn't it? We're just recording, we're tracing it. It depolarized, but just not enough here. And here it did, so it continued. And again, this is what you're going to see is if I get enough of a change here, it will cause a change here, which will cause a change here, and so forth. Okay? You're getting this cascade. It's like dominoes in a sense, in a very simple sense, but that works, right? So if not enough, nothing happens, no action potential. If generate enough change in the potential, right, this millivolts, I've hit threshold, and it's going to spread. Right? So this ligand-gated is going to result in a voltage-gated. Cause and effect. All right? So again, as I've already mentioned, you guys, a little bit about these action potentials. They are all or none, right? If threshold, right, the amount of change, again, threshold's the amount of change that has to occur, and then we measure that change in millivolts, right, is reached, there will be a charge reversal. In other words, it'll go from being negative to positive, as we've said. And the action potential will happen. All right? And I said the action potential will always be the same magnitude. You're not going to go above plus 30. There are rules, and this has to do with physiology, but for now, just you won't go above plus 30. A stronger stimuli only increases the frequency, right? Not the magnitude. I'm going to send it more often. So I never stop letting you know. It's there, right? Okay. Proverbial thorn in your side, it's always there. Okay?
What happens if you go over 30?
You just can't. Again, this is limitations, yeah. Its limitations have to do with the physical, these guys and the channels themselves. So, and again, this is, a lot of this is dictated by laws of physics or at least very strong theories of physics. Right? This is, but yeah, it's, 2 millisecond window, it happens very quickly, right? All right. So we're not giving you the units of time across here, but this is a very quick change. Which again is our benefit, right? Because that means we can respond to some stimuli very quickly. The faster we can send information, the better we're going to be at survival, right? So now on this, the theme of this slide is to tell you why your action potential is not going to go backwards. Right? That's really what it comes down to. So why, when I'm moving down that axon, right, and I get, here this, right, localized potential reaches, all these changes that occurred here that we're talking about, result in, here at the hillock or initiation segment, okay, this term's fine, right, there, we get enough change, we've created enough change with the ligand that the voltage gated ones here are going to start opening. And we're going to spread it down here. Well, if each part of the neuron is influencing the next part, why wouldn't what happens here, right, if we're going this way, then affect what was back here? Why couldn't it do that? Yeah, but why does it only move one way? That's what this slide's about. That's what I was trying to get at. And that's why, right? We call it the refractory period, and there is an absolute and a relative refractory period. All right? The simple way to look at this is the absolute is you absolutely cannot send another action potential. Cannot be generated. All right? And this is because you can't open the sodium gates. That's a simple way to say it. Just you can't. And then relative is that the sodium gates would open, but really what's happening is the potassium ones haven't closed yet, so you're kind of fighting, right? All this potassium leaving with all the sodium entering. So it's very hard to reach threshold in that circumstance, right? Because the potassium gates are still open during the relative. But in the absolute, it's simply because you can't, right? The sodium channels haven't reset, right? So, and that's why we talk about these inactivation and activation gates, which again, I'm not going to use on a test question. So you're not going to see anything on a test that says you tell me about these gates. But when we talk, this is why, as Katie observed correctly, it really only moves one way. Now you can change this circumstance with drugs, but it's not going to end well for you, right? In fact, there's some toxins that organisms produce which actually influence this and cause you to be hypersensitive in your sodium channels. And therefore what you do is you spasm, which makes you very easy to catch as a prey item. Yeah. So, but under normal functioning, right, because these sodium gates are not going to respond for a short period of time, which we call absolute refraction, or refractory period. And then, again, the relative means, look, you've got a lot to overcome. You could get an action potential, but the reality is you haven't closed these potassium gates yet as you're opening these sodium, and you got positive leaving just as much as positive trying to come in, so the change, there won't be a change. And you need change to get an action potential. Yeah, Kale?
Are the axons always thicker than any other of the entry, like any of it?
I don't know that to be true.
Because it always looks like it's just like a logical place for electricity to always go anyway, for the direction.
I don't know that it's true, honestly. I could just put it that way. I just don't know that that's true.
Yeah.
They do vary. I mean, nerves clearly vary in their widths. So I don't know if there's a standard axon, standard dendrite width.
Yeah.
I just don't know. All right. So again, that's the easy way. I don't see myself again writing a question because this is, like I said, it's going to be. I really want you to just figure out what depolarization is, repolarization is. This is just, again, know that we're using these specific gated channels to do this. But this might not be so evident right away to you. But this is really the answer for why it won't go backwards. Okay? And it's very interesting because we talk about this in cardiac muscle as well. Cardiac muscle has a very, very, very long refractory period, which is why you can't get, right, contractions right on top of each other. So this is a safeguard in cardiac muscle to prevent fibrillation, honestly. So there's a lot of different applications for that. But, all right, so the bottom line is, again, you're going to proceed along and you're going to go in one direction. All right, so and here's Here's moving down the axon. And again, you'd have this local change that occurred because of voltage, right? So these would be your voltage gated. And it would cause a change here, which would cause a change here, right? So you just move down this long structure, right, that we call the axon. Just beginning at the hillock, lots of voltage gated sodium and potassium channels, and we just keep going. Again, the absolute refractory period means that the sodium channels, which have just been opened, cannot open again, right? And it moves in one direction down the axon. Right? That's really the point there. We're just going to keep moving. And again, we're going to add some myelin here, because if we have myelin, well then we actually, this change here influences the change here. It doesn't have to change everybody in between, which means we can move faster, basically, with myelin, right? So, but we'll look at that in the next couple slides. All right, so again, hopefully fairly simple rules. The hard thing to relate to is, again, is you don't see these ions and you don't think daily basis usually in ions. So here, when myelin is around, it speeds it up because, again, we don't have to cause all the change in here. So that means, again, that these nodes have to be pretty close together. And they generally are. They're generally aren't going to be more than a millimeter apart. I mean, really pretty close. Right? Because what change is occurring here, these voltage gates, is influencing this one. So if they're too far apart, that would be a problem. Right? If this was solid, that would be a big problem. Because the myelin would even prevent the movement of the ions, wouldn't it? So by having these bare places along here, and these, of course, you know, this is the peripheral nervous system, right? Because these are Schwann cells. Right? This is not the central nervous system, because the myelin is in the central nervous system. That is what your white matter is in your brains, right? And your spinal cord that you guys have in front of you. But I'm just saying, when you look at that cartoon, you know this is the peripheral nervous system, right? So, all right. So there's gaps in this myelin sheath. The ion channels exist, so they cut a jump. They call it saltatory, and it works really well. And we know it works really well because we see the consequences when it isn't working in people, when their myelin is being destroyed because of a disease process, and they lose their reflexes, right? They're more likely to fall. They can't control some of their bodily functions as well, right? And so we see the consequences when you don't, right? So graded potential. So now that we get the rules, okay? So don't get flustered about language, okay? Please don't. Okay. So now that we know the rules for the basic communication from neuron to neuron, we have to back up and remember, just like your model shows you, that neurons are receiving a lot of information, right? On the previous cartoon, I gave you an example where there were 3. But we know that neurons exist in what we call pools, neuronal pools. So neurons can be in huge, millions, possible, 100,000, let's say, easily, of, right, connections between. There'd be a lot of potential for that, or a lot of ability, if you keep my potential to my electricity, right? A lot of ability for communication. And that's important, right? Because we want to receive awareness of important senses, don't we? We want to be aware of the world we live in. And that world is complicated with all kinds of things coming in all the time. And your brain that integrates and controls responses, right, is going to have to sort that out, right? It's going to have to figure out, do I need to do something about that? Is that a bad thing? Is that a good thing? Right? You see what I'm saying? So this is where we start to be complicated. All right, so again, this is just reiterating, right? Local changes in the membrane potential, right? This negative, positive, right? Start with the dendrites. The change correlates with the strength of the stimulus, as I already said, right? The stronger the stimulus, so brighter light, right? Greater force, more weight, like that mosquito, right? Right, strength of the stimulus. You've got a couple choices. It's not infinite. That's the good news, right? You don't have all these, all these things I have to track. I've got a couple choices because we are not infinite in those channels, right? So if sodium or calcium enter the cell, right, I'm going to depolarize, aren't I? Because that's rules. They're high outside. They're both positive. If they enter, I will depolarize, won't I? I mean, that's going to happen, all right? And that, I would say, would result in an excitatory. I've caused a change, and we're just going to say it's exciting, right? Because we talked about excitability, right? If potassium or chloride leaves, as Katie told us, right, then the inside becomes even more negative. And I'm hyperpolarized, which means it's harder to meet my threshold, isn't it? I'm getting further away from that magic minus 55. Much harder to reach my threshold. So we would say you were inhibited. So because you actually can be getting signals of all four of these at once as a neuron, you got to figure out what you're going to do. And so we're looking at this kind of idea. Again, we've got these depolarizing potentials coming in, but they don't hit threshold. This one does. So this one would cause an action potential. All right, they're not showing you the action potential. They're just measuring the change here right now, just the change here, okay? All right, and then we have these hyperpolarizing, which can go minus 90. They might even go lower. I'm not sure how low they can go. All right, so they're showing you a tracing because we could track this. Again, this isn't something we're making up. We could look at, right? And we're after, remember, the graded potential is the word we use for the stimulus. So here we had enough to actually change things. In this circumstance, we didn't. All right? Okay? So that's it's just saying, think about this. That's all we're saying here is, not trying to make your heads hurt anymore. So we, of course, call this summation, right? So what you're going to do is going to depend on all this incoming, information from your other neurons. So postsynaptic potential, okay, what does that mean? That means what happens here. Because these are presynaptic, aren't they? So in here we do talk about the presynaptic and the postsynaptic, and all we're saying, just like we did in chapter 10, is that you're either before or after the synapse, right? So we're after the synapse. What's going to happen here in this neuron that's getting information from, 1, 2, 3, 4, 5, 6, 7, 8, 9 other neurons? What's it going to do, right? So postsynaptic potentials formed in the receiving, right, either get excitatory or inhibitory. There isn't any choice. You're either going to increase or decrease. That's why I said, really, at the end of the day, in physiology, that's what it comes down to. All right? So then they look at this again. These are excitatory postsynaptic potentials, inhibitory postsynaptic potentials. That's just the abbreviation here. I won't use that on a test question. All right? I'm just explaining the figure. So that's what I said. So here we are. We've got a probe in this, right, receiving neuron, and we're just tracking this. So at this point, we actually made threshold, right? We got our change. But I say, what does a cell to do? Because you sum all these. These neurons have to sum. And the cell body, right, is receiving, right? And it has to determine whether or not an action potential be generated. And what it basically goes on, if we hit threshold, then we're excited and we're going to go. But all along here we didn't, so the impulse is inhibited. Which we do need both, by the way. All right, so it's not, oh no, it's always good to send. You ask anybody in chronic pain and they probably really want to inhibit, right? Oops. All right, so synapses you're already aware of, but the chapter reminds you that, okay, here's if we looked very closely at this, again, that's that space between the two. Here's that neurotransmitter. We looked at acetylcholine being released, okay? So this, again, we made all our changes in this presynaptic neuron, and the action potential is coming all the way to the end, and some event down here is going to cause this to be released, okay? Well, it turns out as that voltage change, right, the action potential gets down here, there are specialized calcium channels here. Yeah. And so calcium, if the action potential reaches the end of this presynaptic neuron, it will open calcium channels. Calcium will enter, and the entering calcium will cause these vesicles, right? These are all just packages of neurotransmitters to fuse with the presynaptic axon and release a neurotransmitter. Right? I say here, right, there can be electrical transfer, but that's not very common. So you don't have to retain that if you don't want to. I'm just being, that is a possibility. What we are going to talk about is to say our focus is really the chemical, as we talked about in the muscle and motor neuron. It's acetylcholine, right? So I say, in a synapse, here's your parts, right? You got to have the presynaptic part, really has to have calmed down this other axon. You have some neurotransmitter. You have the cleft, which is the actual space, right? You have receptor proteins, so they have to be there, or this means nothing, right? And this, again, can be what's the problem if you don't have a receptor. Your postsynaptic, we've got to make our localized changes because of this binding and opening channels here. So these would be ligand-gated channels. And if I get enough of a change here, I'd spread it. All right? Now, we also remember that since this axon is telling this cell body and dendrites of this other neuron what to do, that this is the chemical messenger, right? The neurotransmitter is called the chemical messenger. And as long as it's around, this one can still respond, can't it? So I said to you guys in chapter 10 that in the cleft itself, associated with the neuroglia or the neuron itself, are enzymes that destroy, right? Acetylcholinesterase was the one we talked about. Destroy. So we're to stop the communication, that's what we're talking about here. Somewhere in this area would either have to be an enzyme to destroy the neurotransmitter, or there'd have to be a cell or the neuron itself that would, take it back up, right? Endocytose, because it's exocytosing here, isn't it? It would have to endocytose it here, or a neuroglia cell could do that, right? But I'm going to keep on making change if the neurotransmitter stays there. And so I said, that's why we take advantage of that when we treat depression. We use these selective serotonin reuptake inhibitors. It's in the name, SSRIs, right? We want to keep serotonin around. So that drug blocks the mechanism of the serotonin being returned. And as long as the serotonin is around, it's going to cause a change in this other axon, which is going to lead to, again, perceived happiness or whatever, stability. I don't know. I'm still waiting to take my first antidepressant. So, all right. So now transition. Every semester I put that off, right? So hopefully it'll still be this semester, you guys. All right. So transmission of that stimuli again. So here we are. Here's our calcium channels in the cartoon. Here's our synaptic vesicles exocytosing, leaving, right? Ligand-gated receptors here. It's just slightly different color. And then we enlarge this to see, oh yeah, sodium enters. Okay. So this is just walking me through, because I said, what's really important in us at this point is to get the cause and effect, to get this order of sequence of events, so that we know we can show some understanding. So I said, action potential arrives, right, causes these voltage-gated calcium, what I'd already mentioned to you guys, right, open, calcium moves in, causes the release of neurotransmitter to diffuse across them, right, the neurotransmitter, again, doesn't enter the cell, right, it's going to bind to a receptor. It's not supposed to enter the cell. We don't want it in the cell. We want it just to cause another event, which is opening the channel, right? And it just depends. But, as I said here, it's kind of limited. You're going to see sodium, potassium, chloride, calcium, possibly too. But whatever that neurotransmitter is going to dictate which channel opens. They'll all be a ligand-gated channel. Right? So this is where we get complicated, because there's lots of different neurotransmitters, and I'm saying that we have choices at what they're going to do. All right? So you are not going to be tested on this. See, I can be nice. Right? You're not going to tell me about the difference between the cholinergic, the biogenic, neuropeptides, and amino acids. This is a courtesy slide. Just like I told you, I'm not going to have a question and ask you about the difference between absolute and relative refraction or refractory periods. This is important because knowing this, we have manipulated a lot of people's lives, and generally in a good way is what I'm hoping. All right, so I'm hoping it's a good way, but only time will tell us that. There are many different neurotransmitters that the body makes, and we're not through figuring out all of them yet. All right, but of the ones of their chemical signature, what they look like molecularly, to understand them, we've grouped them because we need to compare and contrast. Remember, because they have this power of exciting or inhibiting action potentials. So we have the cholinergic system, which is what acetylcholine belongs to. All right. All right. You're going to see that the autonomic nervous system uses this as well. Please don't be surprised. All right. There were two types of receptors. You do not have to know this. The book mentions this, but okay. Nicotinic is what you've been looking at so far. There's also this muscarinic, and they do different things. So that there's added complexity, right? So amino acids is another group of neurotransmitters, and that includes glutamate, right? Aminobutyric acid, which we call GABA, and glycine. Yeah, Katie?
So for the first one there, nicotinic, does that have to do with like nicotine?
It was probably first described as such, but it's been expanded, right? Biogenic amines, so they're made from amino acids, but they are missing a component, like the carboxyl. Just to let you know, serotonin falls into this. It comes from tryptophan, which is why people always say, well, eating turkey, right, gives you that sense of satisfaction. You got to eat a lot of turkey to get this, right? There's just not that much in it. So we've done the studies, trust me. But serotonin does come from tryptophan, right? Dopamine and epinephrine and norepinephrine are from tyrosine, all right? And these are also called adrenergic, is another term we use because we associate them with the adrenal glands, because that's where they were, I think, first described as being produced by adrenal glands. Neuropeptides, right? So, again, in the name, small groups of amino acids, short chains, and enkephalins. So these are really our pain, our pleasure, if you will, and enkephalins. Substance P, endorphins, all right? And I'm just saying that there's at least 5 different enkephalins and 31 different endorphins, and it's because pleasure is a strong motivator for activities by all species. And I just remind you that these chemicals are put together by that, because this was actually one of the more questions on the quiz as well, when I asked you guys about the rough endoplasmic reticulum. A lot of people told me myelin for some reason. It's, of course, it's chromatic filler substances. It's just you're getting used to the words. So please spend some time to get used to the words, all right? So made and transported down the axons, right, and stored there. That's all those vesicles you saw, stored there. So they can be used when the right voltage change occurs. All right, effect is complicated. It depends on the receptors. Again, this is why this is a courtesy. All right, courtesy to you guys. And when acetylcholine binds as nicotinic, the postsynaptic is depolarized. If it's muscarinic, it might be hyperpolarized. So we can get the same neurotransmitter giving opposite effects. It depends on the receptor. All right. Glutamate is generally excitatory. Glycine and GABA are hyper, so they're inhibitory. So we're just giving you some rules here, but not ones I'm going to test you on. Okay, they're important things as you move forward, because again, clinically we manipulate people's nervous system a lot, right? Biogenics don't generally change electric potential, but they affect transcription and other metabolic events. So again, the limit, this you should know, because we mentioned this in chapter 10 as well, is you're gonna limit the response because of an enzyme like acetylcholinesterase, or you're going to take that neurotransmitter up to stop the effect. You want an on-off switch that's rapid, all right? So, but you've got to remember, it's those neurotransmitters that are really key to the communication between these neurons. So how am I going to control communication and get the effect I want, all right? So that it's just going to be a simple type question about, what is it that are how do we limit, again, this here, how do we limit the effect? All right, last slide. Yes, it is. Yay, we did finish the chapter. Woo! All right. So, again, since I'm continuing the trend here, this is not what I'm going to ask you about on a test, but this, again, shows you the complexity and why we're not there yet in our complete understanding. I don't know if we'll get there in my lifetime, but all right. So there are two classes of receptors on top of being, the nicotinic or muscarinic. There's also just two broad classes. And the inotropic, which we mentioned, again, when we talked about ligand-gated, that was acetylcholine. And then there's the metabotropic. And these are complicated. These are the messenger systems that rely on G proteins and phospholipase C. And these cause a lot of internal. So this is good to make you aware of, because when you start talking about hormones, is your first chapter, endocrine system in AMP2. We will talk about taking all these chemicals, yeah, sound familiar? Neurotransmitters, chemicals, hormones, chemicals, right? We'll talk about taking all these chemicals and putting them into two broad categories that we just say steroid or non-steroid. And we've mentioned the steroid, so the non-steroid generally do this. So again, what are we talking about? We're talking about a powerful chemical that influences another cell. That's what a hormone is, right? And so this powerful chemical could be a cell really close by, or it could be a cell like in the hormone system, endocrine system, quite a bit away. But the mechanism is similar. So again, we call these guys first messengers. Hormones are also neurotransmitters, are called first messengers, and so are hormones. They bind a receptor, because that's what a non-steroid would have to do. It can't enter a cell. These neurotransmitters can't enter the cells, all right? And they activate groups of proteins to get change. So it's a fascinating area, but again, it's not really. We're an introductory class, so I'm not holding you guys accountable. Please understand, I'm very serious about that. But it's in your book. It's an important thing to read and kind of contemplate, especially because you might have some familiarity, real world experience with this stuff.